Here we are, finally. Got through MRI physics last time, and now we can talk about the fun stuff, fMRI. So start with, what are we measuring? Well, we're measuring the signal from hemoglobin. So hemoglobin is the oxygen transporter molecule in the blood, and it's a special molecule because it can hold up to 4 oxygen atoms on it. So anywhere from 0 to 4, basically. And when it's bound to oxygen, especially fully bound to 4 oxygen molecules, it changes its molecular configuration. So it's basically a different shape depending on whether it's oxyhemoglobin, that is with oxygen on it, or deoxyhemoglobin. And it turns out that oxy and deoxyhemoglobin actually have different magnetic properties, which is why it's relevant to fMRI. Sure why I have, oh, I see. So I have this little animation that shows how it's changing its configuration, bound and not bound. That's kind of it doesn't so much matter for our purposes how it changes configuration, so much as that it does change configuration. And in particular, deoxyhemoglobin is what we call paramagnetic. And what that means is that it's somewhat magnetic, so the configuration is such that it has some magnetic properties. And so it has its own little magnetic field around it. And what that means in your body is that where you have concentrations of deoxyhemoglobin, you actually have localized disruptions in the magnetic field. So if you think about last time, everything in MRI is based on knowing the Warmore frequency of the tissue that you're trying to image. And doing things like slice selection we do by creating magnetic field gradients that actually change the strength of the magnetic field systematically across the brain so that we can tune our radio frequency pulse to a particular Larmor frequency and excite a particular slice. And so when you've got something in there that's disrupting the magnetic field, the effect of that is to actually sort of disrupt the MRI signal quite locally. So I'll get to the pulse sequence in a minute, but the original studies that demonstrated what we call the BOLD contrast, or blood oxygenation level dependent contrast, were done in rats in the late 1980s, early 1990s, originally in test tubes actually, and then in live animals. And so in this figure, what you can see, let's look at the top. So compare A and B, so top and bottom on the left, and then also top and bottom on the right. And what do you see different about those images between the top and bottom in each pair? It's not really actually a better signal-to-noise ratio. I think you're on to something, but that's not quite the right answer.
Like
Christina?
more like veins in the top versus the bottom. You can see a better picture.
Right, and you see them as what? Dark lines, right, exactly. And so in this experiment, what they did is they had rats breathe in either normal room air, which is about 20% oxygen, or air that was hypersaturated with oxygen. And the difference is with normal room air, you have basically a higher concentration of deoxyhemoglobin. When you have hypersaturated air where there's so much oxygen, you basically saturate the hemoglobin, and so you have much less deoxyhemoglobin because it's all oxyhemoglobin. And so what you're seeing in the top is those black lines, as Christina said, the blood vessels. That's being caused, so that's when they're breathing room air. Bottom ones are when they're breathing hypersaturated and their blood's fully saturated. And so when there's more deoxyhemoglobin, then you see these black lines, and those are being caused by disruptions in signals, so basically signal drop-off. So we'll get to the pulse sequence in a minute, but basically this pulse sequence is sensitive to magnetic field strength, broadly speaking. That's a bit of a fudge, but what you're seeing is that the black lines are because you've got deoxyhemoglobin, and that's causing disruption of the magnetic fields. So where all the pulse sequence parameters are set, assuming a particular magnetic field strength there because of the strength of the scanner itself and the magnetic field gradients, the deoxyhemoglobin is actually changing that magnetic field quite locally in the vessels. And so the signal drops off faster there than it does in the other parts. So just as you use these things like T1 and T2, T1 relaxation, T2 recovery, as ways of getting contrast between gray matter and white matter and CSF in structural imaging. In fMRI, you're using this contrast between oxy and deoxyhemoglobin to get a functional image. And the parameter that we're manipulating or playing with in the MRI sequence is actually the T2 star decay. So remember last time, so the T1 recovery is where you send the RF pulse in, it tips the net magnetization vector into the longitudinal, the transverse plane, where you're actually measuring the signal, and then it slowly recovers. So that's your T1. Your T2 is that when you tip it into the transverse plane, you're also causing phase synchronization. So all those atoms that are precessing are all synchronized, and they're precessing together in synchrony, right? And then when you turn off the RF pulse, you turn off the music, they stop dancing to the beat, and they all sort of fall out of synchrony. So that's the T2 decay. T2 star decay is basically that, except accelerated by local magnetic field disruptions, in our case by the deoxyhemoglobin. So the top panel here is basically showing that dephasing. So this is like we're looking down into the transverse plane. So when things are really nicely in phase, you see that over on the left. As they dephase, you can see the different protons end up with different sort of phases of precession. And what you're seeing on the bottom is basically how the signal decays. So this is the actual signal that's being read out from the MRI scanner. Not worrying about spatial encoding here, but just the sort of core oscillating signal. And you can see that the amplitude of that signal decays, and it decays quite quickly. So T2 star decay is basically T2 decay that's accelerated by the presence of something that's disrupting the magnetic field, and in this case it's the deoxyhemoglobin. So that's the signal we're measuring, and we'll come to the pulse sequences a little later, but basically we're using pulse sequences that are particularly sensitive to this T2 star decay. And so I showed you those images from the rat brain that were published in 1990. Very quickly after that, they realized that this could be exploited to do human functional imaging using what's called intrinsic contrast. So a contrast that's intrinsic to the body, as opposed to having to introduce something. So up until then, basically, functional imaging was being done using PET, where we'll talk about that in a few weeks, but PET involves introducing a radioactive tracer into the body. And so there it's an extrinsic contrast because you're introducing something else into the body. Here they were able to demonstrate a way of getting functional imaging without having to introduce any chemical into the body to trace it through. And the first fMRI reports were actually, there were 3 different labs that were working on this, and all published the results in 1992. 2 of them in the same issue of the same journal, Proceedings of the National Academy of Sciences. And this is from one of them, I think this is from Kwan et al. Regardless, in this study, they had people lying in the MRI scanner, and they were just showing them checkerboards, so these sort of displays of on and on, dark and light squares. So a very basic visual stimulus, and that was on for a period of time. You can see here it's about 70 seconds, and off, and alternating just off and on. And the logic there was that brain areas that are sensitive to visual stimulation should show changes in this BOLD signal that correlate with that, right? So when the stimulation's on, activity should go up. When the stimulation's off, activity should go down. And brain areas that aren't doing visual processing shouldn't show that systematic fluctuation. Whatever they're doing, whatever noise is in there, it shouldn't be systematically correlated with the stimulus presentation. And sure enough, what you can see here in the brain activity over time is, sort of brightness at baseline, which is just sort of we can ignore that. That's sort of everything's referenced relative to that. Then the off period, and then when the visual stimulus is on, you can see that it's particularly bright in the occipital lobe, so the back of the head. So here we're looking down at the top of the head, front is at the top, and back is at the bottom. So occipital lobe is the visual cortex, so it makes sense. Turn the stimulus off, the activity, or the signal, I should say, goes down, and then you turn it on and the signal goes back up. And then this is just a time course plotting the actual signal values from one pixel inside that occipital cortex area. And so they did this in the visual system, and one group did it, or 2 groups did it in the motor system as well. And there they just, and this is, the very early days of fMRI. So we didn't have sophisticated stimulus presentation kinds of things. So the motor task was just you would hear somebody over the intercom say, "Start tapping your fingers," and you just tap your fingers on one hand, and then so like it could be tap your right fingers and do that for 30 seconds. Stop. Okay, stop. Then tap your fingers on your left hand. And we know where the motor cortex is roughly, and we know that it's crossed. So we know if you're tapping your right hand, we should see left motor cortex activation. Tapping your left hand, you should see right motor cortex activation. And that's indeed the systematic pattern that they saw. So all of the experiments were very simple and just designed to prove basically stuff that we already knew existed in the brain. So occipital cortex should light up for visual, motor cortex should light up for motor activation, and that's what they found. So quite revolutionary, first time anybody had done this with MRI, and as I say, it opened up whole new possibilities because up until then the only really spatially resolved imaging was PET, which involved radioactivity, which was obviously quite limiting. Okay, so this was from Quantital. Oh yeah, and here you're just seeing the motor cortex stuff. So this is now a coronal view, so it's like you're looking at the person face on, and you can see one hemisphere is lighting up and not the other because they're just tapping with one finger. All right, so all that is cool, but there's one little catch here, which is, you might have, and if you read the chapter, this isn't going to be surprising at all, but think back to the rat images. When they were breathing room air, there was actually black lines, right? So the signal was lower because of deoxyhemoglobin. So if we think about this, increased neural activity means the neurons are doing more. So they should be doing more metabolism, right? They're more active, their cells, and they're demanding more energy. Metabolism depends on oxygen consumption. If we're consuming more oxygen, that should mean that we're increasing the amount of deoxyhemoglobin, right? So we're taking more oxygen out of the hemoglobin molecules to feed the cells. And so that would predict that you would actually get a decrease in the BOLD signal. Like in those rats, you should see black areas where there's activation because you're consuming more oxygen, so there's more deoxyhemoglobin, which means more disruption and the signal should drop off faster. But that's not what we see. You saw those bright white images, and you saw in the plots that the signal was actually going up when there was activation and not down. And so on the one hand, it was kind of cool that you see these areas light up, just kind of as you would intuitively expect. On the other hand, it wasn't quite clear what the mechanism was that was causing this because it's like the response from the brain seemed backwards to what anybody would rationally predict. And so it led to a whole field of study around cerebral metabolism and what actually is going on in the brain when neurons become more active. And this actually it's fairly well resolved now. I mean, like anything in science, there's still open questions, but it took a decade or more to really nail down the answer and clarify what was going on. But basically, if we sort of step back, so the brain consumes about 20% of the total oxygen in the body, even though it's only about 2% of body weight and gets about 11% of the output from the heart. And so this is at rest, I should say, irrespective of any sort of visual stimulation or anything else, right? And that's, of course, in spite of the fact that people say, oh, you only ever use 10% of your brain at any one time. Those are people who are usually trying to sell you something about how to use more of your brain. In fact, all of our brain is active all the time, right? We're awake, we're conscious, even when we're unconscious. Our brain cells are still firing, they're still, maintaining homeostasis and so on. And that consumes a gross amount a disproportionate amount of the body's resources in terms of oxygen. And in fact, a lot of that is driven by the sodium-potassium pumps, which you can see sort of schematized here, right? So anytime a neuron fires, it causes changes in the balance of sodium and potassium ions across the cell membrane, and then it has to use these pumps to rebalance those. So if you think back to like intro neuroscience, the action potential, this is the mechanism that's in place, and it's why there's a refractory period after a neuron fires. It takes a while for these pumps to re-equilibrate the cell membrane potentials before it can fire again. And it turns out that this is consuming the vast amount, or the vast proportion of the resources in terms of metabolism. These pumps take energy to work, and that's, they're going all the time, and that's consuming a lot of energy. And then the action potentials themselves. So, and I should say that it's not just during action potentials that these pumps are active. They're active all the time to maintain that homeostasis, and then they sort of ramp up a bit when there's an action potential. So the action potentials are maybe like a 10% on top of that. And then neurotransmitter recycling. So after neurotransmitters are used, they're sort of spit into the synaptic cleft and they do their magic. Then they have to get taken up by glia or the neurons themselves and sort of recycled so that they can be used again. And that's another 12%. And then the rest is sort of miscellaneous other stuff. And this is, again, still just the brain at rest. So it's already doing a lot in terms of demanding oxygen and using that to maintain its homeostasis. So when people started looking at this, and they were using different, so this is largely focused on human studies. There have been some animal studies as well. But the question was, in humans, what's this energy being used for? And then above and beyond the resting state, what's happening when particular brain areas are stimulated and they increase in activation? And there were, broadly speaking, two prominent hypotheses, colorfully illustrated here. So one is the anaerobic hypothesis. So anaerobic means without oxygen. And so like when you sprint, that's when you're going anaerobic because you're putting demands on your muscles that are far in excess of what they can generate in terms of energy just on the basis of oxygen metabolism. And so there's a whole sort of separate chemical pathway that I'll talk on in a minute to support that very short duration, high energy consumption sort of thing. And then the other is aerobic, which is using oxygen to do the metabolism, illustrated by SpongeBob and friends doing aerobics. So the question was for the brain, when, so having established that the brain's already consuming a lot of energy at rest, so that's all, so the resting state is already an aerobic model, which is the way most of the body works most of the time, which is why we breathe oxygen, right? So if it's already doing that, the question was when small groups of neurons, proportionate to the rest of the brain, show increases in activity, is that kind of like a sprint? So they switch into like an anaerobic mode to support these brief increased levels of activity, and then they go back down to the sort of aerobic level, or is it all based on oxygen consumption? And so I apologize if anybody has flashbacks to biochemistry and nightmares and that sort of thing, but this is basically the chemical pathway, biochemical pathway by which oxygen is converted into energy in the body. And it involves a number of steps, including glycolysis, so breaking down glucose or simple sugars. So the main energy source in the brain is a molecule called adenosine triphosphate, or ATP. And so this whole process is how do we create ATP out of sugar. And basically the aerobic process involves oxygen, so you can see oxygen molecules going in there. Don't worry, the TCA cycle and all this is not on the test, but just to sort of understand. So if you go through the aerobic pathway, you're consuming oxygen, producing carbon dioxide, and you end up with 34 ATP molecules for one molecule of glucose. So it's highly efficient. It generates a lot of energy molecules from the glucose and requires oxygen. Over on the right is the anaerobic pathway, which is sort of much, at least here, shown much more simply. And the critical thing is it doesn't require oxygen, but it's far less efficient. And so you end up producing far less ATP, so just 2 rather than 34 from the anaerobic. So that's why our bodies tend not to use it, and just use it in cases where the oxygen supply isn't strong enough. So if you start sprinting, your leg muscles are demanding too much energy, your lungs can't keep up, and so that's when you start to feel the burn in your legs. That's the lactic acid that's produced as a byproduct of the anaerobic metabolism. And so the thinking here, and I've sort of glossed over some details, but there was some work that was done using PET imaging. And in PET imaging, the nice thing is that you can actually quantify the amount of oxygen. So you can use radioactively labeled oxygen, quantify how much oxygen is being metabolized. You can also use radioactively labeled glucose and track the amount of glucose metabolism. And so you can basically quantify the extent to which the brain is engaged in aerobic versus anaerobic metabolism by looking at the concentrations of glucose and oxygen. sort of together. And the sort of short story of all of this is that the actual ratio of oxygen to glucose in the resting brain, as shown with PET imaging, was 5.5 to 1, whereas if it was going solely through aerobic metabolism, it should actually be 6 to 1. So it seemed like there was actually some anaerobic metabolism already going on in the brain, even when you just have people lying in the scanner not doing anything in particular. And so that was interpreted as suggesting that the brain is already sort of maxed out in terms of at rest. It's consuming as much oxygen as it can, and there's sort of no bandwidth to increase neuronal activity levels using aerobic metabolism. So the thinking was that the brain had to rely on anaerobic metabolism for these short transient increases in activity that we would get in sort of cognitive neuroscience studies and brief stimulation and so on. So that was one hypothesis. But some other researchers took a different tack, so they weren't looking at humans, they weren't using PET imaging. Instead, they were looking at animals, specifically cats, and using a form of optical imaging, invasive optical imaging, where you basically create a craniotomy. So you open up the skull, expose the cortex, and shine light onto it. And the light has, it's light of a specific wavelength, so like a laser kind of thing. And you measure the reflectance. And so by shining light of different colors, you can actually quantify the amount of oxygen in the blood and the amount of oxyhemoglobin, and the amount of deoxyhemoglobin. Because if you think about blood, like your veins are kind of more purpley, whereas oxygen, your arteries are more red. So oxygenated hemoglobin is a more bright red color, and deoxygenated hemoglobin is a more purpley color. And so just by reflecting light of different wavelengths to different colors off the tissue, you can quantify the oxy and deoxyhemoglobin separately. Whereas in fMRI, it's all sort of conflated. You're just getting this one signal. So by doing that and then stimulating the animals, so they expose the visual cortex of the cats under anesthesia, prop their eyes open, stimulate them visually, and they got a response that you see here, where when they measured the oxyhemoglobin, they saw sort of a delay and then a big rise and a big increase in oxyhemoglobin. And for deoxyhemoglobin, they saw this interesting pattern where there's initially a little rise and then a big dip. And so the interpretation of this is that, the original aerobic hypothesis is there's blood coming in, oxygen's going to get sucked out of those hemoglobin molecules used for the neurons, and then there's more deoxyhemoglobin in that area. What these data are showing us is that's kind of true. So that little peak there in the deoxyhemoglobin is showing that, yes, initially after stimulation, there's a slight increase in deoxyhemoglobin, suggesting there are more oxygen molecules being pulled off of it and used. But there's this huge increase as well in oxyhemoglobin. And that's not being consumed, so there's this sort of flood of oxygen into the activated brain area that's far in excess of what that brain area is actually going to consume. And they described it with this phrase, watering the garden for the sake of one thirsty flower, right? So you've got some neurons that are slightly increased in activity, so the metabolic demands go up, and the body's response is to flood that area with way more blood, way more oxygenated blood than it actually needs to do its job. And so that's where this BOLD signal comes from, that even though we expect that there's going to be an increase in oxygen consumption, we actually see a signal that tells us there's way more oxygen there than there was before. And it's because we're flooding that area with more oxygen than it needs. The reasons for this, it's physiology and evolution, and we can't really know. The speculation is really that it's sort of just to make sure your brain doesn't run out of oxygen, right? So it's to make sure that the neurons, that you don't suffer in terms of brain activity just because your neurons are more active. So there's essentially a reserve there if the neurons get even more active. And so that was fairly compelling evidence that the metabolism, the increases in activity associated with neural activity are actually aerobic. And it's just that there's this big flood of oxygenated blood, which is why you don't see the decrease. Okay. So that was fine up to a point, and then people kept digging into exactly what's the mechanism, because all that tells us is there's a flood of oxygen. It doesn't tell us really why or how that occurs. So a number of papers. So originally this came out of work by Pellerin and Magistretti, who sort of came up with this model, and then over time, other people, including notably Takano et al., did studies to prove it. So the complicated name is the Astrocyte-Neuron Lactate Shuttle Hypothesis. And the thinking here is that, as trying to illustrate here, so we've got neural activity, so we've got ascending neuron, receiving neuron, a synaptic junction there, and those synapses, so these are excitatory neurons, so they're using glutamate as the primary neurotransmitter. Turns out glutamate, I talked about neurotransmitter recycling before, glutamate's recycled by astrocytes, so a particular form of glia. So as neural activity occurs, glutamate gets spit out of the sending neuron into the synaptic cleft, activates the receiving neuron, and then that glutamate's kind of floating around, and the astrocytes take it in and deactivate it, so change its chemical structure slightly, and then they actually have processes that feed it back into the neurons. So that's the neurotransmitter recycling cycle there, is the astrocytes take this glutamate up, change it around, send it back to the neurons to use again. In doing that, they rely on anaerobic glycolysis, so that glutamate recycling step involves anaerobic metabolism, which is part of why there was evidence for anaerobic metabolism in some of the earlier studies. And within the astrocytes, that uptake of glutamate is actually a trigger for a whole different chemical cascade inside the astrocytes, involving calcium and COX-1 and then prostaglandin. And the other interesting thing about these astrocytes is not only do they have processes that sort of, and by processes I mean anatomical structures, not chemical processes. So they've got structures that are near the neurons and sensing the glutamate, but they also have these end feet that wrap around the capillaries. So the capillaries are the tiniest blood vessels that are between the arteries and the veins. And that's really where the oxygen perfuses out of the blood and into the surrounding tissue and gets to the neurons. So the astrocytes actually touch these capillaries, and the prostaglandin that's produced in there is a type of hormone that actually causes the capillaries to dilate. And so the effect is that as glutamate levels increase, the astrocytes detect that, and that triggers a chemical cascade inside the astrocytes that causes the blood vessels to dilate. And that's why you get this flood of oxygenated blood into that area, is because the capillaries are actually getting physically bigger, and so more blood can flow through and saturate those areas. So that seems to be the process as well as we understand it right now. So these astrocytes are really sensitive to neural activity levels by way of glutamate and the influence in the diameter of the blood vessels. Does that all make sense? More or less? Okay. So that's the chemical process, and I think the really important thing to take away from that is, besides the fact that the BOLD signal reflects in this gross increase in oxygen in activated brain areas, the main sort of takeaway here is how indirect the relationship is between changes in neural activity and changes in the fMRI signal that we ultimately measure, right? Because neuron fires more, sends more glutamate out. That goes to an astrocyte that is doing some anaerobic metabolism, and then there's this whole sort of chemical cascade of recycling glutamate, triggers calcium, triggers COX-1, triggers prostaglandin, triggers dilation in the blood vessels. So there's many, many steps in between change in neural activity and increased oxygen to that area. There's still a relationship, which is why fMRI is actually a useful tool, because it does reflect changes in neural activity. But it's quite an indirect relationship. And so you can appreciate that there are a lot of different things that could affect what we call the coupling between changes in neural activity and changes in this BOLD signal, right? So different drugs that you take, chemicals in your body, and so on, if they affect any part of that chemical cascade, could affect the fMRI response, even if they're not directly affecting the brain response. So obviously some drugs and chemicals will affect neural firing rates. That's going to have one set of effects. But then there are other ones that just might affect how responsive, like, your blood vessels are, right, to that effect, how much they're going to dilate, how, changes in there. So in principle, at least, there's stuff that could affect the fMRI signal that have no actual impact on changes in neural activity, but they're affecting something in between, in that pathway between the change in neural activity and the actual blood vessel dilation. And that's really important to understand because a lot of the time people don't fully appreciate that. And it has some impact on how you interpret fMRI data and some of the analysis approaches I'll talk about next class. What was the other thing I was going to say about that? Oh yeah, the other take-home message there, and this actually I'll come back to, is that what that means is that what fMRI is really sensitive to is not so much the activity level of the receiving neuron as the activity level of the sending neuron. And so if you think about how neurons are structured, typically you've got sort of the cell bodies that receive input and then an axon that goes out, and sometimes the axon goes to another part of the brain and sends signals there. And so the sort of short story there is that the fMRI signal is actually more sensitive to the input to a particular brain area than it is to the spiking output of that area. So it's more like it's sensitive to the postsynaptic potential changes at the synapses than necessarily what the neurons do with that information, which is not unlike EEG, where it's really all about the postsynaptic potentials, right? I think the big difference, so it matters in both. The big difference is that with fMRI, we have very high spatial resolution. So we see changes locally in a particular brain area, and we say, oh, there's changes there. But that's really primarily reflecting changes in the information coming into that brain area, necessarily, rather than necessarily what it's sort of sending out. Whereas with EEG, because everything's sort of spatially all mushed together, it's almost more of a moot point, because with EEG, it's more like we've got these little components that are biomarkers of particular cognitive processes, and we can see changes in the size of those components or presence, absence of those components that we infer a particular process is happening or not. We're a little less concerned about the precise localization and so on. I'm going to come back to that in a little bit. All right, so the hemodynamic response function, and this is maybe sort of two sides of the same coin, but it's a little confusing maybe that we have two terms. So we've got BOLD response and HRF for hemodynamic response function. So the BOLD response is basically the effect that blood oxygenation changes as a function of neural activity. The HRF is just this actual line here. So it's like the time course of the BOLD effect after you see a stimulus. So basically what you see is at time 0 on this plot, that's when a stimulus occurs. And this is a response to a very sort of transient brief stimulus. So goes on, goes off, and you get this kind of response from an active brain area. And critically, the HRF is quite slow, and this is the inherent temporal limitation on fMRI data altogether. So if you look at the time axis, it's typically like 6 to 8 seconds for the BOLD response or the HRF to peak after stimulation. It actually takes 2 to 3 seconds to start really rising, 6 to 8 seconds to peak, 10 to 12 seconds to come back down to baseline, and then often there's this undershoot before it fully recovers to baseline. So basically from a single stimulus, you've got about 20 seconds for the whole HRF to play out and things to come back and stabilize at baseline again. So that's super slow when you consider that when we look at EEG, ERP, in the first second after stimulation, you can see like a P1, an N1, a P2, an N2, an N400, a P600, right? You can see all these different fluctuations in brain activity that we know are coming from different brain areas and actually reflecting different cognitive processes. When we're looking at fMRI, all that gets smushed together because all that different sort of electrical activity has all happened a long time ago by the time anything is measured with the fMRI response. And that's a huge limitation of fMRI, is it's very good at the spatial resolution, the where, but it's really terrible at the when question of when different areas are activated. And I'll come back to that. Actually, this sort of helps illustrate it. So this is from a study where they just threw a bunch of people in the scanner, did a very simple task, and measured empirically what the HRF looked like from each individual in activated brain areas. And so the black is what they call the canonical, or sort of the model HRF. So kind of like what I showed you before, just not with that undershoot. So a little oversimplified. And then each other colored tracing there is the response from one individual. And so you can see that the peak, so there's 5 seconds. It's a little hard to see with a big round spotlight there, but you can see that some of them peak fairly early, well before that canonical. Some peak after the canonical. They're different in their height. They're different in afterwards as they go down. Some of them show a pretty big dip before return to baseline. Others just kind of taper off. So there's a high degree of variability between individuals in the timing of this response. Yeah.
What would be meant by a negative bold signal change before there's the stimulus? Because the stimulus is at the 0 time, right?
Yep.
So a couple of them are almost at -1,
yep.
when there's a stimulus.
Yep.
Why would that not be set to 0?
That's a good question. In this case, so I guess the basic answer, and this is something I was going to come to anyway, so it's good that you asked. Bold is not inherently quantitative. So you get a number out of the MRI scanner at every time point in your fMRI scan. Those numbers are arbitrary, and they depend a lot on sort of the scanner, and the scanner baseline itself actually drifts over time. And so, if I put you in the scanner and then I put Rhiannon in the scanner 2 hours later, even the first scan before anybody's seen any stimulation, the actual numbers I get off the scanner are probably going to be quite different. And so, and this is something we'll come to more next class in terms of experimental design and that sort of thing. But it basically means that the numbers you get have no independent meaning. They're just sort of some arbitrary number. And what matters is how that changes with stimulation. So that's why, even in those original studies, they're looking for that sort of on-off alternation, right? It's not enough just to throw somebody in the scanner and throw visual stimulation at them and say, oh, now there's X% oxygen concentration or something. It's not quantitative, it's all relative. And so here, the bold signal change is done in %. And so they've got some baseline, which probably means that before the stimulus came on, they were scanning, they've got something going on. Why it's a little below there at time 0, that suggests that maybe there was just sort of inherent fluctuation in things, yeah. But it sort of illustrates the fact that those numbers are kind of arbitrary. The % signal change is a way of trying to get something quantitative, because at least now we're measuring relative to sort of the baseline, whether that's immediately before, or you've just sort of averaged over the entire scan, and then everything is sort of relative to that average. Could be something like that. Okay, yeah, but the main point is that there's a lot of variability in the shape and the timing of the bold response between individuals. And it turns out even within an individual, if you look at the timing, the shape of the HRF, it varies from brain area to brain area. And it can vary a bit within a brain area depending on the task that they're being given. Obviously, tasks might affect the magnitude, but it can also affect the timing. And so what that means is the timing information in fMRI is not very useful. So you can imagine, yes, it's slow, but we could potentially interpret, this purple one, which is a bit later compared to, say, there's a yellow one there that's earlier. You could say, oh, well, if there's a one-to-one relationship in terms of timing between neural activation and peak of the bold response, even though it's delayed, we might be able to interpret that as, say this was all within one subject and purple is one brain area, yellow is another brain area. You could be tempted to say, oh, the purple area activated later because its bold response is later. But that, again, if you think about that whole chemical cascade, that could just be that the purple area, it was slower to recycle the glutamate. Or the, sort of prostaglandin release to the capillaries was a little slower, and the capillaries reacted more slowly or something like that, or there are less capillaries in that particular part of the brain compared to another part of the brain. There are lots of different reasons why these things could be different that have nothing to do with the neural activity. And that's really important to understand because fMRI is so indirect. All right. Now, that said, it's not voodoo. I've already sort of showed you at a molecular level the relationship that we understand between neural activity and bold response. But this group led by Nikos Logothetis in Germany showed this in vivo in monkeys. And so in this experiment, which was huge, it was published, I don't even have the date there, early 2000s, I think, a huge tour de force in terms of the technical development that was required. Because what they wanted to do was do single-unit electrophysiology in monkeys inside the MRI scanner. So they wanted to put electrodes directly into the monkey brain and record from them while simultaneously doing fMRI to understand how the actual neural electrical activity related to the bold signal changes we see with fMRI. And because MRI is using a big magnetic field and then these gradient magnetic fields that are changing as we do the acquisition, that's all going to induce currents in your electrodes. Because remember the right-hand rule, any current induces a magnetic field, but any magnetic field induces a current. So all these switching magnetic fields that are going on with MRI scanning are going to induce currents in any electrode, in any wire that you have, and then the radio frequency pulses as well. So it was a lot of, and without going into all the details, I show this mostly just to show you that it's super complicated. But this is the electrode itself, and it had all this stuff on it, kind of like MEG where you've got the active shielding, so you're measuring all the electromagnetic waves that are hitting the MEG scanner in the room and trying to subtract those out. They did this on a small scale by having sensors on the electrodes that were measuring the impinging magnetic fields. And then there was a lot too. Basically, this is sort of showing you, and it's a little hard unless you can sort of appreciate what the final signal should look like. But the raw signal is very high amplitude and dominated, like you can see these sort of alternating spikes and so on, which is all related to when the magnetic field gradients are on. And so it's a matter of trying to measure all of that stuff and subtract it out until you get just the residual electrical activity that's coming from the neurons out of that. But they managed to do it. And the other part was training the monkeys because, like I mentioned the cat studies where they could anesthetize the cats and then prop their eyelids open. That's kind of harder with monkeys in an MRI scanner. So they actually had to train the monkeys to be awake and go into the MRI scanner. And for whatever reason, they kind of have to crouch down with their head in rather than lying. I guess monkeys don't like lying on their backs. And so a lot of work just to get this data. And then just doing simple visual stimulation. And what you see here is the occipital lobe. So that's the midline. So this is like the left occipital lobe here. And this thing here, that sort of line, is the electrode. And all that stuff around it is just sort of the fixative that holds the electrode in place there. So they've got an electrode stuck into the occipital cortex. And then over on the right, you can see the bold fMRI activation superimposed. So this basically just shows us that they were getting bold signal from the part of the occipital lobe where they were sticking the electrode in, which is a good starting point, right? And then the question is, how do these signals relate to each other? And in fact, that electrode, it was a multi-electrode array, and so they were able to record both single unit activity, which is basically action potentials, so that's where you poke one neuron, and you're recording it spiking, but also multi-unit activity, or local field potentials, which is kind of like the micro EEG. So it's where the electrode isn't stuck directly in any given neuron, but it's just measuring the ambient electrical field around it in sort of the extracellular fluid. And so that's picking up the postsynaptic potentials from all the neurons that are sort of locally around that. So they're able to measure both the spiking output of the neurons in that brain area and also the local field potentials, which are the input to that area, and then the fMRI bold signal. And so what you see helps start with the legend down here. So the bold signal's the sort of fleshy colored signal there. Local field potentials are the black line. Sorry, I said things wrong before. So the multi-unit activity is actually the spiking. You can ignore the SDF. And the big question is the bold signal, A, is it related to either of these things? Hopefully yes. And B, if it is, which one is it more closely related to, the spiking or the postsynaptic potentials? And in general, sort of the short answer, so the contrast is better when I do this, isn't it? So the bold signal you can see is delayed, so the sort of hard to do this and also point. So the bold signal's here, so it's delayed relative to the onset of the stimulus. You can see that the multi-unit activity, so the spiking, goes up right away, that green peak, and then the local field potentials go up as well. They're sustained a little longer than the spiking, and then they sort of gradually go back down. So again, it emphasizes that the bold response is slow compared to the neuronal activity, which we already expected. But also what they found is basically a higher correlation between the bold response and local field potentials, so the postsynaptic potentials, than the correlation between the BOLD response and the spiking. So again, like what I was saying before, the BOLD response is mostly or more dominated by the local field potentials in the input into the brain area than it is by the spiking output of that brain area. But it is correlated with both. And that's not entirely surprising, right? Because the input to the brain area should be correlated with the output of that brain area. Make sense? All right. Okay, so the next question is, given all this, in humans, where we're not sticking electrodes in the brain, and more broadly, how do we measure it? And the way we measure fMRI typically is using a type of pulse sequence called echoplanar imaging, which was developed by Sir Peter Mansfield, for which he won a Nobel Prize in Physiology or Medicine in 2003. So he was English. Paul Lauterbur was American, also an MRI physicist. They shared the Nobel Prize because the Nobel Prize was basically sort of for a whole body of work that they did jointly in developing the technology for MRI. But Mansfield was primarily responsible for coming up with the echoplanar imaging pulse sequence. And what's particularly interesting about it is that he invented it decades before anybody could actually do it, because the MRI hardware at the time wasn't capable of doing it. And so it took decades for the engineering to actually catch up with the sort of theoretical side of what he had invented. The Nobel Prize was for the slice selection, developing the method of slice selection using gradients and the whole approach of using magnetic field gradients and also analysis of RF signals. And I mentioned, actually, remember last time I talked about the Russian who invented MRI, but nobody believed him and he never got any credit for it. In that article, there was mention of this other guy, Damadian, who felt that he should have shared in the Nobel Prize. And one of the key reasons that he didn't was that his implementation of MRI didn't have gradients. And so that whole process of coming up with the gradients to do the spatial localization wasn't part of what he developed, and that was key to the Nobel Prize, and also key to echoplanar imaging. So here's the EPI Pulse Sequence. So if you remember back to last time, there are a lot of elements of the EPI Pulse Sequence that are already familiar and shared with structural MRI sequences. So the RF pulse goes in, excites the tissue, and at the same time you apply a slice selection gradient so that you're exciting only one slice and not the whole brain. The key difference is that EPI is a way of scanning really quickly. So what I described last time for structural imaging, you would do the slice selection and then you do one phase encoding step. And so along one dimension, so say our phase encoding dimension is front to back in the head, you'd create a gradient of some steepness along that, create that phase roll, remember those sine wave gradients, right? So create that sort of stripy pattern. So you create one sort of phase roll step, and then do your frequency encoding to read out the other dimension in your slice. And then you have to do another RF pulse combined with slice selection, and then a different phase encoding gradient to get the next line, and keep doing that repeatedly. So to get one slice, you have to do multiple RF pulses, multiple phase encodings, read it out, and that all takes time. The idea of echoplanar imaging is after a single RF pulse, you can complete your whole slice. You can do all the phase encoding steps and all the frequency encoding steps. And the way they do that is essentially not any different from the structural sequences, right? You still have to create a magnetic field gradient and then read that signal out, create a magnetic field gradient, read it out. But where the conventional sequence sort of does that slowly over time, here we do the phase encoding step, turn on frequency encoding, then we do another phase encoding step. And that's really what's illustrated here is that, so phase encoding is the vertical dimension in our K-space plot. So normally you would just fill one of these lines per excitation. Here what you do is after one excitation, you do the phase encoding gradient for one step, read out the frequency, which is the horizontal dimension, and then you actually switch the phase encoding gradient. So the slope was one way, now it's the other way, and then you do the frequency encoding again, and that basically reverses its direction, and it goes back and forth. So practically what's happening is rather than sort of turn on a gradient for a while and then read it out, you're rapidly switching these gradients in different directions. And that was actually the part that was difficult engineering-wise, is it turns out to be quite challenging from an engineering perspective to create these gradients and then reverse them and flip them very quickly. It also creates some other issues. So with these EPI pulse sequences, if you ever have an fMRI scan, one thing they will tell you is don't put your hands together like this. And that's because you can actually, when you do that, you're creating a closed loop. And these rapidly switching gradients can actually induce an electrical current. Again, right-hand rule. So you can actually, it wouldn't probably be harmful, but you could actually feel like peripheral nerve stimulation and tingling in your nerves because this rapid switching is going to induce these currents, and they're going to be flipping back and forth. So from a practical perspective, there's some interesting things. It makes the sequence very loud and very, if you have a conventional MRI sequence, it sounds sort of like mrrr, mrrr. An EPI is like beep, 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 beep. So you can sort of, even when you listen to it, you can hear that it's scanning very quickly. But it's critical for fMRI because for fMRI, we want to think about that bold response I'm showing you. Things are changing on sort of a second to second or every couple of seconds. So with a conventional structural MRI, when I talked about the pulse sequences, I didn't mention the timing, but a structural MRI will take anywhere from like 3 to 12 minutes to scan through the whole brain. An EPI sequence will take about 2 seconds to scan through your whole brain. So super fast, and that's where the real advantage lies. Now the advantage in the structural one, and the reason we don't use EPI for structural imaging, is that the structural imaging gives you much better signal-to-noise ratio, better contrast to noise, which means that the white matter, gray matter, all that is much clearer. EPI images look much more fuzzy. They're not good for structure. They're just good at picking up this T2 star kind of contrast. And so what you see here is actually a structural MRI, that's sort of the darker image, and then the EPI image of the brain superimposed on it, which is the sort of white stuff. So you can see compared to, you can't see a lot of the brain there, but the structural is pretty rich in terms of its anatomical detail. Whereas with the EPI, you can kind of tell, maybe more when I don't shine the light on it, there's a bit of gray-white matter contrast. So you can kind of tell there's like a brain there and some of the structure, but it's not nearly as clear or as crisp as in the structural images. The other cost of EPI is what's being illustrated here and what the arrows are pointing to, which are called magnetic susceptibility artifacts. And that's because you're switching so fast, these things are very sensitive to small disruptions in the magnetic field. Because again, all of this switching is predicated on this assumption that we've sent in an RF pulse, we've done our slice selection gradients, we're using all these gradients to very precisely specify the Larmor frequency at each point in the brain. And all of our image reconstruction is based on being able to predict the Larmor frequency at each point and pull the signal out for that. It turns out that especially where you've got different types of tissue, and particularly air versus tissue in the head, you've got big differences. So air has very different magnetic properties than CSF or brain or any sort of biological tissue, and that causes localized disruptions. And so you get these artifacts, which basically are signal loss or holes in your brain image near areas of pockets of air in your head. And the pockets of air in your head are mostly your sinuses, so under your frontal lobes, which you can see in the top ones here. So you can really see how, here's the EPI image. There's like this little divot out of the frontal pole, and the orbital frontal cortex isn't totally covered there. And then the other one is your ear canals. So the inferior temporal lobes above your ear canals also show these kinds of distortions here. And so that's one of the downsides of EPI, is you get these artifacts, and they're parts of the brain that are actually harder to image with fMRI than other parts of the brain. Orbital frontal cortex, frontal pole, and inferior temporal lobes. The inferior temporal lobe ones are particularly frustrating for some people because that's where the hippocampus and the amygdala are, which are a big interest in memory and emotion studies, especially. Now, these aren't necessarily insurmountable issues. This is, the ones I've shown here, I selected ones that showed particularly prominent artifact. So more modern pulse sequences have ways of trying to compensate to some degree. It also turns out that it matters how your slices are angled. So these were done with straight axial slices, so they're sort of horizontal, slices through the head. When you're setting up your scan, you can actually tilt your slices so they're not necessarily straight horizontal slices. And so if you have some idea of sort of where the pockets of air are, like you want to avoid the sinuses, you can actually tilt your slices in such a way that they actually sort of line up with that boundary better, rather than slicing through it. They're sort of parallel to it, and that can help with the image quality as well. I'm not sure what the best current practice is for hippocampus. I know for a while, instead of doing axial images, people were using coronal images, because again, just having the slice in a different plane, it cut through the air pockets differently and caused less distortion. Yeah.
So out of just curiosity, if you're to run like an fMRI on someone like HM at the bilateral lobectomies, would it just be chock-full artifacts and it wouldn't all be all that useful? Or
well, in that case, and in any case of brain resection, wherever the brain is missing, there's CSF. So it's a liquid-brain interface, so that's much less of an issue than an air-brain interface. So the issue would be more if, for whatever reason, somebody had extra air pockets or something sort of outside the skull cavity that the CSF is contained in, but sort of near there, yeah. Sometimes you get distortions around, like if somebody's had neurosurgery, you can tell because there's sort of differences there. Usually, if that's the case, it's more because they have like metal pins or something. Like they'll use metal screws to attach the skull back after they've put it back in. So there are other issues there. The other thing that you can do is there's a different type of pulse sequence you can do that actually basically maps out the magnetic field. So when you've got the person's head in there and their head is distorting the magnetic field in their own sort of idiosyncratic way, you can run that field mapping sequence and get a map of exactly where the distortions are. And then in software, you can actually use that to sort of correct for those imbalances. Mostly that doesn't, the issue is that these sorts of things are missing data, right? And so if you sort of correct, all you're doing is sort of warping the data that you do have to sort of match the way the brain should look. You can't recreate data that wasn't there in the first place. So it helps more with aligning the functional images with the structural images, rather than necessarily allowing you to image areas that would otherwise be difficult to. There's more engineering that's required to do that imaging right. Okay, just a sort of a brief mention, because this is less and less common. There's an alternative functional MRI pulse sequence that was used for a while quite widely called spiral pulse sequence. And it was invented sort of in parallel with EPI, but as a way of being less demanding on the hardware. Because even when people started using EPI sequences, only certain MRI hardware had the specifications to be able to do it. And so the idea of the spiral pulse sequence is, rather than doing this rapid back and forth switching of your gradients, you're actually adjusting the phase and frequency gradients sinusoidally together. So you can see that in the pattern here. Remember, the EPI one, the phase and frequency encoding steps are like these squares, right? So they're sort of up and down, on and off. Here, they're fluctuating slowly. So basically what that means is that, rather than sort of being sloped one way and rapidly switching to the other, the gradient's just sort of slowly changing over time, and at the same time, the phase encoding gradient is sort of slowly changing as well. And so from a hardware perspective, it's gentler and easier to implement. It actually has different artifacts than the EPI sequence. So it was also sort of potentially a way of getting around if you had issues with the particular EPI-specific artifacts. You could try using spiral imaging as a way of imaging those hard-to-image areas at the expense of some other kinds of image distortions. The issue with spiral was that it just, it's kind of like, standards in any field. Something catches on and becomes the dominant standard. Spiral was developed by Gary Glover, who's an MR physicist at Stanford. He had a research agreement with GE, who's one of the MRI manufacturers. And so, and at the time, I think GE didn't have their own EPI pulse sequence. So if you wanted to do fMRI using a GE scanner, it was easier just to get Gary's pulse sequence, plug it in, and use it. But then GE developed their own EPI pulse sequence, and other companies like Philips and Siemens, who make MRI scanners, never did spiral. They always just focused on the EPI. And so the EPI kind of became dominant, and the hardware caught up to the point where spiral wasn't really necessary anymore. So it's probably something that you would more see in older MRI papers at this point.